Well, welcome everybody. I just wanted to thank everybody for coming, and I just wanted to introduce Evzenia Chepkova. Holds a PhD in math from Purdue University and specializes in statistical learning theory, machine learning, high dimensional data analysis. Her research explores dimensionality reduction, proportion of high accuracy models, and generalizability in connection with randomness and data set labels. And our workshop today is on dimensionality reduction in action. So take it away, Evzenia.
Thank you. Okay, so I just wanted to double check if everybody can see the notebook or is able to access it. Let us start then. So today we're going to discuss high dimensional data sets. Examples of high dimensional data sets are images. The dimensionality is given, for example, by the number of pixels. So even a small image, 64 by 64 pixels, already has 4096 dimensions. As human beings, we're used to processing information from one dimension, two dimensions, three dimensions. Spatial objects around us, we're able to somehow intuitively analyze. But already when it comes to four dimension, it's really hard for us to visualize, to imagine what's happening in the data, and to see if there's a pattern in the data or similar signal. Another example, maybe a textual data set, genomics, the number of genes can be in 25,000, or audio data sets where the dimensionality is given, for example, by time samples. So the plan of the tutorial today is to take a look at the curse of the dimensionality at first. We have two main problems with high dimensional data. Visualization, we would like, as data scientists, we would like to see how the data looks like in order to create some image available we can extract from the data, and clustering. Often we would like to separate the data into groups that are meaningful, groups that represent something. And as we will see, curse of dimensionality makes this problem of clustering hard because it depends on the distances between the points, and the distances behave strangely in high dimensions. Next on our agenda is random projection. Random projection is a very quick way to reduce dimensionality, and we will cover two surprising facts about random projections, some mathematical theoretical guarantees, as well as some new ideas about how random projections can be used for clustering, for example. After that, we will cover more traditional techniques like VCA, linear reduction. These are techniques that allow us to interpret what we're doing with the data easily because they're linear and we can exactly say what we're doing. But we will also cover some nonlinear reductions, and we will look into autoencoders, which is a neural network that helps us reduce dimensionality of the data, and we have certain control over the architecture of the network for our purpose. And we will end the tutorial with other nonlinear mappings that are useful mostly for visualization, Riemann and TSNE, that are good for preserving a local as well as global structure of the data and allow us to see what's happening in the high dimensional data set projected to the dimensions. So if you're going through the notebook, please follow the first cell. Now also go to the notebook over here. This is just some basic libraries in Python that we're going to use. My background is in mathematics, so the code that I will show is not going to be too complex, or I think it may be really, for many of you, very intuitive, but we are here mostly to discuss what properties the high dimensional data have and what can we expect of them, kind of building an intuition about high dimensional data sets. So our main focus today is looking for clusters, and the clusters do not have a very precise formal definition, but intuitively, we are looking for points that are concentrated close together. So we're looking for points that are closer together than the other points. For example, we have a iris data set here. You can call the next link. This is a very famous, a very simple data set that contains 150 points of 4-dimensional data. We're going to plot the second and the third dimension of the first 100 points and see just a little visualization of what we kind of expect the data to look like. So in our mind, we want to find points that are close together, like the green points here and the blue points over there. And we imagine that it will behave like this in the high dimensional setting as well. But the problem with high dimensions is that the space in between the points grows exponentially fast as the dimensionality increases. So points become separated from each other. The space becomes sparse, and it is it is somehow realized in the fact that for a large range of data sets, if we take our data set and then we take another point, our query point, let's say, and we measure the distance to the closest point in the data set and compare it to the distance to the furthest away point, they become indistinguishable. So in our low dimensional data sets, everything is nice and close together, and you can see that the nearest neighbor is much more closer to the point than the furthest away neighbor that belongs to another cluster.
Just need to share your screen so it shows up on the Zoom.
Okay, sure. I'm sorry. Yeah, I didn't do that. Yes, but in the high dimensional data sets, often for a lot of distribution, it happens that there is one point, then there's nothing for a long time, and then there's another point. Then there's nothing for a long time, and then there's another point. And then we still need to cluster that type of data, even though the distances do not behave the same way as we intuitively expect them to.
Does that paper show the derivation of that equation, that inequality?
Yes, yes. And it also mentions the range of the distributions that has this problem. And it's an old research. I think it's from 90s, but the people tend to somehow forget about things that were discovered before. And what I wanted to emphasize in this tutorial today is that we have to be careful about what we're doing. We have a lot of methods to find nearest neighbors, but then maybe by doing that, we're not achieving what we actually set up to achieve, because data sets can be different. So this paper says that for a wide range of distributions in probability, when dimensionality grows to infinity, you can express it in different ways. But one way to express it is that the maximum distance to the query point becomes very close to the minimum distance. So it's less than 1 plus epsilon of the minimum distance. And we're going to explore this by sampling data point and tracking the ratio between the maximum distance and the minimum distance. And we're going to use one synthetic data set and 2 real data sets. So if you're going through the notebook, please click on the next cell. This is a little function that measures maximum and minimum distance of a point. That's called a point to the data, which is the data set. Then in the next cell, we're going to compute the ratios between those maximum and minimum distances. We're going to set the number of trials. This is the how many dimensions we're going to try. So we're going to try with dimensionality 1, 2, 3, 4, and we're going to go up to 200. For each loop, we're going to generate Gaussian data set, standardized Gaussian with the 0 mean and identity covariance matrix, and also another Gaussian point that we're going to use as a query point. And the question is, does this hold? Does the sample maximum to the query point divided by the minimum to the query point converge to 1? And as we can see, we can plot the vector of the ratios of the distances in the next image. Here that dimensionality is on the x-axis. The ratio between the maximum and minimum distance is on the y-axis. And we see that it goes down. Since it's sampling, the plot is very jumpy. And we see that it goes under the value of 1.25 with 200 dimensions as maximum. Now, the question is if this holds for some real data set as well. I chose to work with Olivetti data set, which is a data set of 400 images of 40 different people, 10 images per person. And each image has dimensionality of 4096, which is 66 by 66. So try to upload the data set. Does it work? Olivetti faces. We can print the shape. We can see how the targets look like. So you can see that there's a 10 zeros, 10 ones, 10 twos. So the first person has the first 10 images. The second person has the second 10 of the images, and so on. And we can visualize our data set. This is what we have there. Each pixel contains a value between 0 and 1, which is a grayscale value of the grayscale for the complete pixel. On the top, we see the number of the sample, 0, 15, 30, 45. And on the bottom in blue, you can see the label of the person. So person number 9, person number 10 are the last ones in this visualization.
In block 8, those are different intensities of the pixels.
No, these are just target
Assignment.
labels.
Right. Okay, so they're well, the 64 by 64 image. There are 40 people.
40 people, correct.
Okay.
400 images. Each person has 10.
Independent images, or
Yes, it's taken from different viewpoints, maybe different expressions. And we have altogether 40 people in the set. So this is a good question because it actually shows us we have some information about this data set. It's not, this is actually information we're going to use throughout the tutorial. And what we expect from this data set is perhaps that there will be 40 clusters, each cluster representing a person, because a person's image should be similar to the same person's image, much more than to somebody else. So we're going to flatten the data and measure the distance. Okay, here, the query point, we take the first person in our set, and all of the rest we put in the data set. Here we do not exclude the photos of the same person. So 1, 2, 3, over here, the index 1, 2, 3, 4, 5, up to 10 corresponds to the first person as well. So we kind of expect the first point to be closer to those than to the others. But when we plot the distribution of the ratio, we see that it goes steadily down under 4. So it's approximately 3 where we finish. So the ratio that we achieve, I also wanted to mention that the dimensionality here, how the dimensionality grows is that for each sample, we just add 1 more pixel in the loop. So at first we just take the first pixel, in the second loop we take 2 pixels, and in the end we have 4096 pixels. And as you can see, the ratio goes steadily down and it goes under the value of 4. So we now know it's not exactly 1, but we know that this person, all of the points are in the radius of 3 times the closest person, the closest point in the data set to that query point. And we need to fit 40 clusters in that space. So it's not very probable that there will be close clusters put together of people if we have such behavior of the maximum versus minimum distance. I also wanted to show an example of a data set where this does not work that much. So this is a gene expression omnibus data set. We're going to work with these data sets throughout the tutorial. So I will introduce them here and we will come back to them a couple of times throughout. We can download this data set. This one is a very high dimensional. It contains different cells, and cells contain gene expressions. There's 25,000 genes, different genes, and about 1,400 different cells. And these are the counts saved inside the expressions. So please download this file as well. Let's load this as Pandas data frame and see how it looks like. So you can see here that this is a transposed matrix to what we would expect. The samples are in columns and the features are in rows. So we can transpose it and save it as an NumPy array. I'm also going to take a subsample of this data. This I choose arbitrarily, so I will take 4,500 genes. And again, we can measure the distances. Our query point is the first point in the data set, and we measure to the other points in our data set, adding 1 dimension in each step.
So our points now are samples.
I'm
Our
sorry?
points are samples.
Yeah, our points are cells.
Yeah, cells. Yeah.
Yeah. And each cell has some gene expression.
So that's 20,000.
25,000, yes, but we restrict ourselves to
Okay.
4,500. So we create a similar plot here, and we see that the ratio goes from 80 to 45. So it allows the distances are still very different between these points. So what I wanted to say as a conclusion is that some data sets have this property when this maximum distance over minimum distance converges to 1. Those are for sure the data sets where all the features are independent and generated according to the same distribution, like we had in the beginning, the Gaussian distribution. They're all independent based on, because we chose the covariance matrix as identity matrix. So if you have a data set where all the features are independent and identically distributed, then you will have this a property for sure when the distances just kind of lose their purpose. But there is a wider range of data sets that have that property. Not all of them though. And some data sets have some kind of fake dimensionality. So they're in very high dimensions, but they're concentrating on low dimensional manifolds. So then the distances we should actually consider from that lower dimension, and this phenomenon is not going to happen, because even though the data is in 4,000 dimensions, it actually can lay in just a subspace of 3, and all the other dimensions are redundant. But what I wanted to mention next is random projections with theoretical guarantees. There is a lemma, it's called Johnson-Lindenstrass lemma. A version of it says that if you have a high-dimensional data set with a fixed number of points and points, then there exists a mapping to a lower dimension k, so that all of the distances are approximately preserved. So they are disturbed by no more than epsilon, like in this formula. After we project the point, we measure the distance to another projected point, and this distance is going to be larger than 1 minus epsilon of their original distance, but smaller than 1 plus epsilon of their original distance. All of them. So all of the points mapped into a lower dimension k. If the lower dimension k is not too small, there is some bound on it, then all of the distances will be approximately preserved. And this lemma was proved probabilistic. So it was shown that the probability, like you can construct a projection based on random distribution, and that it was shown that the probability of this happening, like no distance is disturbed, was higher than 0. If the probability is higher than 0, then there must be at least one function, one mapping, such that if you use this mapping to reduce dimensions, this property will hold. But even more was shown during the proof. It was shown that with a high probability, this will happen. And thereafter, it was also shown that you can actually fix epsilon, fix tau, and with probability 1 minus 1 over the number of samples to the power of tau, you will get a mapping such that all of the points will be preserved, as long as k is not too small. So this k, the dimensionality to which you project, has to satisfy this lower bound, which depends on the number of points that you have in the data set. The more points, the bigger the k. The epsilon is how much you disturb the data, and tau is connected to the probability of failure. So how sure you want to be that this property will hold. And it turns out that for P, you can use scaled Gaussian distribution or Rademacher distribution, for example. Rademacher distribution minus 1, 1 with the same probability. So then I just wanted to try it out with you and construct a matrix and project our real data set and see how the distances would vary. I also wanted to mention here that this has two consequences. First of all, if the points were concentrated in clusters in the original data set, after such a projection, they're going to be concentrated again because we do not disturb distances much. And second of all, this is a very useful way. First of all, it's quick. We do not optimize anything. We just create a random matrix, and it is data independent. So if you receive data online, like the data is streaming to you, and you don't have the whole data set at the given point, you can use this method of dimensionality reduction by projecting each point, like setting the matrix first and then projecting each point as it comes to you, and it will still, the distortion property will hold. So let us try with a matrix P. One dimension is 100. This is the original dimension of the data we'll have, and we will reduce it to 40, so a little bit less than half. So we need 100 by 40 matrix of Gaussian data scaled by square root of k, and k in our case is 40. let's take a biosample. So we're not taking Gaussian data, we're taking the real data from the biological data set. And we're taking 250 samples and dimension 100. So we restrict our data set to 250 by 100. And we will just multiply this sampled matrix by P. This is our projection matrix. We will get a new data set with the same number of samples, 250, and dimensionality 40. And now the next cell measures the distance between, so here we have 10 trials. We just pick 10 pairs of points at random from our data set, and we measure what was their distance originally and what is their distance after projection. So you can see that there was 331 distance, it became 322, and if it was 97, it became 104. And we can measure the perturbation between the distances by dividing them and subtracting one, taking absolute value. So we can see this 2%, 2%, 7%, 9%. Basically, they're all bound by 10%. We can see that after this random projection, there is no mass disruption in distances. I wanted to mention here briefly what we were doing with my advisor at Purdue, and we were thinking in a slightly different direction, or maybe something that I've already mentioned. If we project like this randomly to one dimension, the distances are not going to be preserved. This is too much. We don't have any guarantee for it from Johnson-Lindenstrass because that gives us the lower bound for K, and it just doesn't hold. But we were thinking that maybe we do not need to preserve the distances, because if the data set happens to be the one that we see in the beginning of the talk, where the distances are just all the points are far away, everything is sparse, then maybe there can still be structure in the data set, but we just cannot access it through the distances. And my advisor and her team noticed that when you project real data sets into one dimension, often what happens is that there is a bimodal distribution like that. This is from this data set, and a lot of times you go through all different projections, you project and you see this. And this is pretty unusual because you expect the Gaussian behavior. And this way you can separate the data into clusters and see what you're gonna achieve. This phenomenon, we try to explain this phenomenon by proposing a model of the data set that actually have this property. And we came up with some cube model. That means that the clusters of the data are not given by their closeness to each other, but by their alignment in some direction. So geometrical structure of the data might be important for the clustering result. Another thought here, which may be important, is that perhaps usually we think about clustering as finding just given clusters. We cluster in two, there's going to be a group here and a group there, but in the data there can be different clusters, and we can project to different directions and see those different clusters based on some different features that the data have. If we have images of people, one cluster may be all the people who wear glasses, and another cluster may be all the people who are women or men, or people who have long hair or short hair, something like that. So there's multiple ways of clustering the data, and we have to be able to also think about that when we analyze our data sets.
Oh, quick question.
Yes.
You might have addressed this, but is the value of K in the Johnson-Lindenstrass lemma best possible, or?
There are different formulations of the Johnson-Dinistrauss lemma. So this is just one that I picked, but there may be closer. Frankly, but if from my experience, what you can prove with math sometimes tends to be far from the practical truth. So maybe you cannot push the proof as much as you want. For example, the training and the test error based on VC dimension, you can separate. You can prove that they will be this much apart, but in reality, in the experiments, we see that they're this much apart. You just cannot prove it, and then you get some experimental results that are far away from the theoretical. And frankly, in this case that we have covered, the values over here, if we plugged in 40 here, 0.10 over here, and 250 over there, this inequality is not going to hold. This is just this would be just too much, and it's going to be hold by a lot. But we can see that when we run the experiment, it's not a very thorough experiment. We just choose 10 pairs of points, right? But we can see that the distances are well preserved, even though based on that inequality, we'd have to pick a much bigger K than we picked. Okay, so I wanted to circle back a little bit and consider a more standard approach to dimensionality reduction, where we use something that is friendly for interpretation. We know what is happening there. And this is PCA, principal component analysis, when we create an empirical covariance matrix of the data. I think there may be an error, and there has to be n-1 in the formula instead of n. So what we do here is we subtract the mean of each data point and multiply each vector by its transpose, and we get a matrix D by D of a sample covariance. And this matrix we can, it's a positive semi-definite matrix, and we can compute its eigenvalues using this formula. So we get lambda 1, lambda 2, lambda 3, up to lambda D. And if we want to project a dimensional subspace, let's say we want to project to 2 dimensions, we will just pick 2 biggest eigenvalues and find the eigenvectors that correspond to them. And these eigenvectors, E1 and E2, for example, they correspond to the directions where the data is spread out the most. So this is, in some sense, the most information that we have about the data. And the other directions may not be that important for us because they don't hold that much information. The data do not vary that much in that direction. So if we have like a blob of data and it does not change in this direction, perhaps we do not even need to consider it, and we'll just project onto this plane where the data change a lot. So I wanted to try to apply PCA on a synthetic data set. And see how it works in this particular example. Here we have 500 samples of 10-dimensional Gaussian data. We will call this data set noise. And then we will have another data set that we will call noise with a signal or signal. And the signal is going to be our noise plus an added vector, which in this case is 1, 0, 0, 0, multiplied by a random coefficient, which I call Poisson, because it's distributed according to Poisson distribution. If we moved all of the points by this vector, the variability of the data in this direction would not grow. So we need to move each point by some random number multiplied by this vector 1, 0, 0, 0. And this way, our data is going to stretch out in the direction of 1, 0, 0, 0. We can plot the data in 3D. So here we're just plotting the first 3 dimensions. We're not reducing the dimensionality. And we see that it looks like this. So the blue points are the noise, and the red points are the noise plus this random number times the vector 1, 0. And what we would like to see is we would like to see if the PCA is going actually to identify this direction 1000 through its process. So we are going to call PCA for two components and apply it to the noise as well as to the signal. Then we're going to print what we get. In this case, for the noise, we get principal components 1.28 and 1.18. And the explained variance ratio is 13% and 12%. So in case of Gaussian data, the Gaussian data are distributed in every direction approximately the same. And you can see in the explained variance ratio that it doesn't change too much. But when we add noise, we get a bigger eigenvalue, 6.5254. And it explains, the first one explains 42 of the variability of all of the data. We can also see if the eigenvector, that corresponds to the largest eigenvalue, has something to do with the signal that we constructed. And we can see that the first index is the same and the other indexes are close to zero. Since it's a finite data set, it will never be exactly the same, but it's very close. And we can visualize what we get after the PCA in 2 dimensions. So these are two different planes. They're not projected into the same plane because we use different eigenvectors, but you can see that the noise is kind of concentrated in a spherical shape, and the noise with the signal is spread out. And this is how PCA actually works with the data. We can apply PCA on real data. Here we choose 3 components. Let's see what kind of eigenvalues and eigenvectors we get. In this case, the first eigenvector explains 24% of the variance, the variability of the data. And we can plot what we get after the PCA procedure. So here I colored the different points according to different persons in our data set. We have 40 persons, and the color corresponds to the true labels in the data set. And the question is maybe if we can cluster after PCA. We'll have the number of, we will use k-means, a very simple procedure for clustering, and we will measure how far k-means is from the true labels. So we actually can see that the clustering is not that good because the true labels and the PCA k-means labels have a small adjusted random score, which is about 0.21. In order for the clustering to be good, it should be at least larger than half for sure. If it's close to 0, then it's not that good. We can also apply PCA on biological data. Here I decided to use the raw data and not to take the logarithm of them, even though that's a practice in the genomic data set that's used a lot. Because the raw data can contain some information that's important for us. If we use the logarithm, we will make the data look more like Gaussian data, and that's not what we want. Here, surprisingly, we can see that one direction explains 88% of the total variability. But this vector is the eigenvector that corresponds to the largest eigenvalue is not very interesting. It has a lot of 0 values, which perhaps is natural for the sparse data set. But we can also plot all of the data points, all the indices that have a non-zero component in them. And if we visualize, we get this type of a three-dimensional projection for the biological data set. Okay, so next I would like to talk about nonlinear reductions. And we're going to use autoencoders for our data sets to reduce their dimensionality. So this is going to be a neural network that approximates an identity function. So we're kind of looking to find fx equal to approximately equal to x. And it has two parts. Encoder that goes from a high dimension. We're going to use it on Olivetti dataset. So I put some dimensions over here. 4096 is what we start with. This is the number of pixels that we have. And we will encode the data and go down to 64 dimensions. And this green is a latent space with a smaller dimensional vector that is supposed to encode the information from the original data set so that we can expand it back to the original dimension and get an output, which is again 4096 dimensional. So this way, we can preserve some information about the data. We can use the data in a smaller dimensionality to perhaps send it over quicker or encode the information that we do not want other people to access. And we will still preserve enough information about the data point, or we hope to preserve enough information about the data point so that we can reconstruct it back. And we have, in this case, we'll have two inner layers in the encoder and two inner layers in the decoder, which is symmetric. And the inner layers will have 1024 nodes and 256 nodes, and they will be all densely connected from one layer to the next. We're going to use ReLU activation function and try to train the network on our Olivetti dataset. So we import TensorFlow, visualize our dataset again, flatten the data. We're going to train, test, separate the data into train and test, and min-max scale our data. The scaler is connected to the activation function in the neural network. In the case of images, it's a good idea to use a min-max scaler, even though in our case it's not necessary because we already have our values scaled between 0 and 1. But if you have some values between 0 and 256, it may be a good idea to scale them to 0 and 1. Our input dimension will be the same as the dimensionality of the data, and the latent dimension we choose 64. We will be able to easily plot it as 8x8 image later on. Thank you. And here is the architecture of our network. Do you have any questions? This is the encoder part. This is the decoder part. Autoencoder connects all the two parts together, and encoder is just the first part that we are going to use for dimensionality reduction. We can compile it using Adam Optimizer and MSE loss, and we can train it. Let me get in on this one. We're going to use early stopping. Early stop is a way to prevent the model from overfitting. So when our training error goes down, the model is happy, but we may not want the validation error to go up while So if the training error goes down, but the validation error kind of stagnates or goes up, then we will stop the training. If we had the 600 epochs in the beginning, we will not go through all of them, but we'll stop at whatever point we need in order to prevent the overfitting.
You have a question? That log plot at the beginning, when you were reducing the dimensions probabilistically, does that inform the dimensionality of the layers that you're applying here? You're,
So you're talking about combining the probabilistic reduction with neural networks?
either way, is that like the dimensionality reducing processes that you had before were showing, how that data was tracking to get to the final point of 40 dimensions or whatever. Could you like map those dimensions to the layers? Like we have, I think 8 layers, I think there was.
Something like that. I think a good idea would actually, people always combine those two, the dimensionality reduction techniques. But one good idea might be to first use random projection. And by Johnson-Lindenstrass, you'll know that all of the distances will be preserved, right? But you cannot project, you cannot reduce too much. So after reducing to, I don't know, 40 dimensions, you cannot reduce more because Johnson-Lindenstrass just says no, there will be no more preservance of the distances. And then you can apply, let's say, autoencoder that's going to find nonlinear mapping on the 40-dimensional data set instead of 100-dimensional data set, which is going to be perhaps faster and easier for you to do. And you already know that you're not going to lose much information because you use the random projection first, distance influence range, and then you can use the autoencoder or some other methods to reduce dimensionalities even further. But in our neural network, we do not apply random projection. And if we were to construct a network that would apply random projection, I mean, you can. I think you will just only have 2 layers, and then you just multiply. You will not be training it even. You'll just have a random weight for each connection between the first layer and the second layer. You run it and you get Johnson-Lindenstrass, but in the form of a neural network, not trained though. Yes.
Is random projection invertible? So if you cluster in your projected space, can you go back to where the centroids were?
Well, you have the matrix that you use.
So you could, rather.
Yeah.
Yeah.
You can take the inverse of the matrix. It's not square though.
So it's positive semi-definite.
Positive semi-definite is the sample covariance matrix, right? When you take the data, you center the data, and you multiply x. This is your data matrix by x transpose. By it kind of looks like x squared, right? So this is why it's but in matrices.
Sure.
So that's why it's positive semi-definite, and that means that all the eigenvalues are going to be non-negative. Yeah, but it does not hold for the projection matrix. Projection matrix is not square, it's like 100 by 40. That's what we have. So I'm not sure about the inverse matrix right now. I think it should be possible. I'm just a little bit stuck. At the moment. Yeah. Okay, so we finished training. We finished training the neural network. And the errors are, I'd say this is very good. So we go, the training error that we finish is even less than 1%, is 0.7, and the validation is 1.6%. And we can hear how, we can see how the errors actually evolve throughout epochs. And this is where we stopped because the validation was just not going down. So now we take the encoder part and we use it to get a version of our data set that is reduced to 64 dimensions. And we can see how it looks like in the real data. This is the faces that we had originally. This is all the same person, and this is a second person. And this is their small dimensional representation. I think the only thing you can notice here is that the 8x8 pictures are much more similar to each other for this person than for that person. But otherwise, I don't know how to interpret it. And our neural network allows us to take this image and spread it out back with certain errors, and we can compare how it looks like. So if we use the whole network and generate the images back, you can see how how different or similar they look like. For some reason, I feel like it often happens that the glasses from people appear on people who don't have glasses. Not in this particular case, but maybe the first person. They kind of look like the glasses appear on them, which may be so ideal. We can try to do the same procedure on biological data, but the errors will be much larger. So here we will take only 64 dimensions of the biological data and again convert it to, or reduce it to 16 dimensions. We'll have a little bit different structure of our network. The first layer, the input will be 64. There will be one hidden layer with 32 dimensions, and there will be the latent dimension of 16. And then we're going to spread it out symmetrically. So we're again going to use the early stopping procedure, and we're going to train our smaller network on this subset of biological data. And as you can see on the errors, it already starts. It starts with a very large error, 100%. The training works well, so the error goes down, but we will see that it's not going to go as much down as for the elevated data set. Any more questions while we train?
In practice, we would normally go to a subset of the genes that are higher variance because
Higher variance.
low variance, I mean, it's gonna be a low,
Not that important.
most of the genes are gonna be very low variance and they're gonna be noise.
Okay, thank you.
Yeah.
I don't have that much practical experience working with biological data. My focus was on theoretical exploration of machine learning, but thank you very much for
So you took what, 40 here, 40, 60?
how many 64 dimensions, but this is
No,
the
how many genes did you take?
64 genes.
Yeah, like there'll be almost very little information.
Very little information.
Yeah.
Oh, yes. So that's why it's not very well trained. But I tried it with more dimensions and
I think you need to throw away the low variance data.
low variance data so that they don't create noise in the training.
That's what we would normally do.
That is-
Yes, it'd be standard.
So here we finish with 26% error and 38 on the validation set. We can also plot it. And see how it evolves in different epochs. Any more questions? Yes.
I came late. I just asked for questions on.
Oh, yeah. Okay. So you said you asked for permission?
Yeah.
To get in?
I just copied it.
I don't see it here, but if you go to GitHub, Telestrom, can you go to this address? Yes, go to dimensionality reduction in action over here. And if you press on this link, there will be a button, open in Colab. Can you see that? Oh. So then you don't need a permission to use that. Great. I guess,
When you use the auto encoders for dimensionality reduction,
yeah.
kind of like PCA is offered with like a kind of some interpretability
Interoperability.
in the variants. So how do you interpret the auto encoder? You're very similar type of way to think about it, or it's just like kind of instructing the dimensionality, and then we don't really know what that means.
More the second way, yes. So this is a usual trade-off between interpretability and some kind of accuracy in machine learning, or at least for now. I do not know many papers that would explain. So there's an explainable AI, right? That means that you take a neural network and you kind of try to see why did it make the decision that it actually made. And there are papers written about it. We had a talk yesterday about it, for example, when you had a picture of a husky and the network decided that it was a wolf, but then you kind of look at what part of the picture it looks at. So for example, that you can do with your neural network. So you can find a paper and there will be code that will allow you to take this autoencoder and see which nodes, which part of the image it made a decision on, something like that. That doesn't exactly hold because that was classification and this is autoencoder, but there are some ways to see which part of the image the neural network used. And for the case of the husky, it used the snow that was behind the animal. So it decided that this was a wolf because of the snow, because the wolves were usually on the snowy place. But yes, so this is a different topic. You would have to go to explainable AI. You'll have to see how people work with it, how they are interpreting what is happening inside of the neural network. But usually the problem is that there's many layers, many connections, and you cannot track mathematically easily what is happening inside, why, how is it projecting, how the weights are trained, why this particular weight is assigned to this particular connection. In linear, like PCA, it is easy to interpret. So you know that you're going to project to this direction where the data is spread out the most. That's exactly what you're making your decision on. And you can set your decision easily. So then you can decide what type of method you need for your particular situation. Well, I feel like with the autoencoder, you have at least some control over the architecture of the network. For example, you can decide how many layers you're going to put, how many connections in between, what kind of activation function you're going to use. But then we're going to cover some additional methods of nonlinear mapping, UMAP and TSNE in the next chapter. And there you can only decide on basically two parameters for training, and then the algorithm does it all by itself. So it's even more, at least I feel like it's even more black boxy than autoencoder. But it gives good results sometimes. I wanted to mention here that you can also use generative adversarial networks to reduce the dimensionality. This is a bit kind of, I don't know if surprising, but the architecture of GANs is not set to reduce the dimensionality like for autoencoders. GANs are created to generate data that would look like the real data set. So they generate synthetic data. And they consist of two parts, generator and discriminator, similarly as autoencoder, but they have different dimensions and different functions. And generator generates new data samples of the original dimension from noise, some kind of smaller latent space. And discriminator looks at this generated point and tries to decide if it's real or fake. So it's a critique that's trained to differentiate between the real sample and the synthetic sample. And this critique, this discriminator, starts with the original dimensionality. In case of Olivetti, that would be 4096 dimensions, and it goes down to 1, which is the probability of this point being fake or real. But you can stop it before, so there is a inner layer which has a lower dimensionality than our original data, and you can use it as dimensionality reduction technique, because the whole network is again trained to preserve some information about the data. So you take a neural network that preserves some information, some kind of interconnections between the points, some kind of information, and then at some point it reduces the dimensionality to a lower space. In this case, it's an inner layer of the discriminator, and we can take it and use it because it can be a good and meaningful dimensionality reduction. And you can look at this biological paper that discusses this topic. Any questions? Okay, so let us go into our last section on linear mapping for visualization. And we will start with UMAP. It's a graph layout algorithm. It's based on the distance between the points in the high dimensional space. It constructs a high dimensional graph and then tries to approximate its structure by a lower dimensional representation, as similar as possible to the high dimensional one. It's a fast algorithm, and it has 2 parameters that we can play with. This is a number of neighbors. That is an estimate of how many close neighbors we expect the data point has in the higher dimensional space. And min distance, which is the minimum distance in the lower dimensional representation of the graph. Original paper can be found here, but I want to show you this visualization first. So if you go to this page, you can see this kind of fun visualization of different animals. And you can switch in between their 3D and 2D visualization. So this is how restricted we are. For us, making anything more dimensional wouldn't be informative. The only thing we can actually kind of grasp is the difference between 3 and 2 dimensions. And the creators of UMAP kind of use this to illustrate how the algorithm works, maybe so that we get some intuition about the results we can expect from this procedure. What I wanted to notice is that looking at the cow, for example, if we just use linear projection on the wall that is behind the cow, if we project it like this, we would get no information about the 2 legs of the cow, right? Because they are going to be squished together. If we project behind the cows behind, then we will get no information about the hind legs. We'll only get the front legs. But if you see what UMAP does to the cows, you can see the projection 2 dimensions, but we can still see 4 legs, a tail, a head, and they're all approximately where they're supposed to be. So the tail is nearby the legs, the head is by the front legs, and we're losing very little information about all of the global and local features of our original data set. The question is, though, how it translates into much higher dimensional objects. Here again, we can see all of the legs.
It's like making roadkill.
Yeah.
Fossils.
Especially the armadillo.
Yeah, especially the armadillo. What it reminds me of is when you skin the animal and then you spread it as a mat.
Very big.
Also similar to that. Okay, so we can try to apply this to our Olivetti data set, and we will set the number of neighbors to 9. So here again, we're using some information about the data that we have kind of on top of the basic expectations. And we set the minimum distance to a very low number. That's just because I experimented with it a little bit, and this was the best. This gave the best output. So it runs for some time. Here also, when I set the random state equal to something for the reproducibility, it runs slower because it cannot use parallel computations that it usually uses. So if you want it to run faster, don't put the random state in. And if you want reproducibility, then prepare to wait a little bit longer, I guess, but it's not too long. I have some estimates here in between 18 and 51 seconds, it should recompute. I think this warning is about what I just mentioned. If you set the random state to something, that it will run longer. And we can visualize what we get as our result. So this is Olivetti data set, and you can see the isolated clusters at some points, like here, 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 and here, and a group of clusters in between. The colors represent different persons from the true labels. So this is actually colored using the real data set, the true labels. And we can see that it's pretty faithful. We can compare it. We can cluster the data using k-means again and see what we get compared to the true data. Don't look at the colors because the colors can be different. Our clusters are just going to find a group of points, but the algorithm doesn't know which is the first person, which is the second person, which is the third person. So the colors do not correspond to each other, but they should be, if there's the same color over here, all green, then it should be all blue, the other picture, and so on. We can also measure the quality of our representation by silhouette score and Davis-Baldwin score. The silhouette score should be close to 1. 1 would be the perfect one. Here it's 65, which is quite good. And the Davis-Baldwin score is should be close to 0. In this case, it's not exactly great. The Davis-Baldwin corresponds to the ratio between within cluster distance and the distances between the clusters. So that's why it should be close to 0. The within cluster distance should be much smaller than the distance between the clusters. And we can also measure how the labels after UMAP correspond to true labels. Here we have 0.54. We can see what the algorithm, what k-means outputs in terms of values for different persons. Some people are very well recognized, like you can see the person number 1 has all 7s. Person number 13 has all 8s. Person number 10 has all 21s. And we can compute how many persons are recognized well in terms of having this value of the same labels larger than 0.8. And we have 17 out of 40 people. We can also try to cluster before we do the projection and see how it compares. Why I wanted to mention this is that it's usually not recommended to cluster after the UMAP algorithm because UMAP distorts the distances between the data points. But in the view of what we've covered in the beginning of the tutorial, it's maybe not such a bad thing to distort the distances between the points, as long as we preserve the information about their being, for example, aligned somehow or being together in a group in a different way. And here we can see that if we cluster the original data set, this is the k-means, and compare how similar it is to the true labels, the value is actually less, it's 0.41, than what we get if we cluster after UMAP. So in this case, applying UMAP and clustering afterwards gives us better results than just clustering the original data set. Okay, we can do the same for the biological data. Here we don't have an estimate about how many close neighbors we have. We choose 5, number of neighbors 5, minimal distance 0.1. And we can plot what we get after the UMAP and a 3D visualization. There's a similar algorithm to UMAP. It's called TSNE. It's very similar in principle, but UMAP is faster and it contains more like shortcuts. Here we can, I wanted to show you some links about TSNE and how to use it effectively. There's a couple of data sets, like 2 parallel lines, 2 clusters far apart, a circle, and I don't know, Gaussian data. And you can try for different level of perplexity. Perplexity is a parameter of TSNE. It's an estimate of how many close neighbors you expect the points to have in your data set. So you can change the perplexity and see how the algorithm deals with different data sets in time. So it's an iterative algorithm, and it stops when it achieves some kind of good result. I also wanted to show you another link where the TSNE and UMAP are compared. I think it's heavily biased towards UMAP being better than TSNE, but you can see a similar plot of small data sets with some easy features and how it's reduced into lower dimensions. Here I would like to notice perhaps one of the most important features of the UMAP is that it also preserves a global structure. So it puts similar things together, but it also puts clusters away from each other if they are not meaningfully connected. For example, in this case, the clothes is separate from the bags, and the bags are separate from the shoes, and the tops are separate from the bottoms. So after this UMAP reduction, you can see different clusters. You can see all of these classes, but you can also see that the distances between the ones that are not similar to each other is higher than if you were to apply TSNE. Okay, so we can try to apply TSNE on Olivetti data set. And visualize what we get. As you can see, it's a much less informative visualization than what we got from UMAP over here or over here. The points are clumped together. I tried to play with the algorithm, but I didn't find a better result. Perhaps you will be more successful if you try to do it at home. If we try to cluster after the projection, after the reduction, then we are not going to get very good results. So here again, we compare to the true labels, and clustering after TS, and it doesn't seem to be like a good idea because the alignment is very low. It's 0.27. This is alignment after applying k-means on the original data, and this is alignment after applying it the new procedure. Okay, we can do TSNE on the biological data and see the result. And then we're going to compare all of the visualizations of the biological data and see how they compare to each other. Do you have any questions?
Still
Or-
testing.
This may be the biggest, the longest cell, the cell that takes the most time.
I have a question. I'm just seeing that it's being recorded. Can we access this?
Yeah, it's gonna be posted or maybe about, it might be a few days before.
Will it be the same for the other?
Great. All the posted from yesterday.
So I hope the goal of this tutorial today was make you aware of some surprising properties of high dimensional data. I guess the most surprising is the random projection that preserves distances and the fact that some distances can become meaningless in high dimensional data sets. And I wanted to just do an overview of different methods we can use for dimensionality reduction so that you have like a toolbox in order when you need to apply dimensionality reduction, and you can combine those methods. For example, you can do random projection first and then apply some nonlinear mapping. You can decide whether you want interpretability in your dimensionality reduction or you want it to be more accurate in some sense. For example, UMAP gives a very good clustering result compared to, for example, clustering after PCA, even though PCA does not distort distances and should be easily applicable without any kind of need to justify it. Okay. So we can visualize a result, which is very, very bad. Apply k-means after all of the biological data, which has number of clusters to be 4 here, and see how the visualization compare. What I also wanted to mention is that, as you can see here, each reduction gives us a very different view of the data. So it's not like there is some ground truth that is preserved after reduction that's easily accessible. Not every picture looks the same, and we have to be actually very careful about what we're seeing, and that this visualization doesn't have to look the same if you apply a different method. We can compare the metrics for the clusterings. This is a comparison between applying k-means on biological data raw without any reduction, and then after reduction, and it is really not very great. And there is 2 silhouette scores and Davis-Baldwin score for our data sets. You can compare the scores and choose the method you'd like the most. but keeping in mind what's happening during the reduction. So this is the end of my tutorial. If you have any questions, please ask.